Well, hello everyone, and welcome to the Neuroblastoma Parent Symposium. It's wonderful to be with everybody today. I am Gavin Lindberg, and I have the honor of moderating our session today. And I will tell you, I'm joining you from my home in the state of Maryland, here in the United States, just outside of Washington, D.C. And also by way of introduction, I did want to mention that my wife, Wendy, and I are angel parents. Our only child, Evan, passed away from neuroblastoma at the age of 7 in 2010 after a 4-year battle that defined courage. And his mom and I continue the fight against this horrible disease in his honor. Now, as you know, the title of our session today is Targeting ALK, Discovery to Frontline Treatment. And our featured presenter is Dr. Yael Mosse. And as many of you may know, Dr. Mosse holds many titles and distinguished honors, too many to list comprehensively here, but I'm going to mention just a few. She is an attending physician at the Children's Hospital of Philadelphia. She is also the Patricia Brophy Endowed Chair in Neuroblastoma Research. She's an associate professor at the Perelman School of Medicine at the University of Pennsylvania. And she is also the director of the Neuroblastoma Developmental Therapeutics Program at the Children's Hospital of Philadelphia. But more importantly than all those titles for me and my wife, Dr. Mosse is a friend. She cared for our son when we were treated in Philadelphia. In fact, my son was actually one of the initial patients enrolled in Dr. Mosse's first ALK inhibitor trial a little over a decade ago. And full disclosure, also the foundation that my wife and I established in our son's memory, the Evan Foundation, has proudly funded Dr. Mosse's groundbreaking work in this area. And you know, we did so because there's just so many heartbreaking challenges and seemingly endless uphill battles with this terrible disease. But the story we're going to hear today is one of progress and one of hope. So I'm very glad that you are all here with us to hear from Dr. Mosse. So one housekeeping item before we get started, I'd like to draw everybody's attention to the Q&A button at the bottom of the screen. This is for the submission of questions during the session. And we are going to get to the questions following Dr. Mosse's talk. And if the button is not showing, just tap or hover over your cursor to bring it up. And then finally, if you see a question that you like, please feel free to give it a thumbs up so that will move it up the priority list for getting posed. So with that introduction, Yael, I want to thank you again so much for taking the time to be with us today and turn the program over to you.
Well, thank you very much, Gavin. It's really an honor to be introduced by you. And indeed, we have become friends over the many years. And you know that Evan's story really motivates me to do better. I'm talking to you from my home in Philadelphia, right outside Philadelphia, and I'm very proud because, as I'm sure all of you know, it's really a historic moment in the United States of America. I immigrated to America myself when I was 10 years old, and just a very proud moment and very proud to be talking with you from Philadelphia. I do want to just disclose that I am a consultant for Pfizer, and actually I can share with you the rationale for why that is a little bit later. I showed this slide when I started in the lab, which was around 2000, so about 20 years ago. I really had no idea what I was doing. I was really the deer in the headlights and had no idea what my journey would bring. But I learned early on that you can see here, in 1967, a pathologist, Jane Chatton, actually, who is from the Children's Hospital of Philadelphia, where I work, described a family where there were multiple disorders, but it included neuroblastoma in 4 siblings. And then it was Alfred Knudsen, who was a mentor of mine and became a friend, and who died just a couple of years ago at the age of 96, who, along with Louise Strong, who is very much still alive, and they predicted that there would be hereditary cases of neuroblastoma, and that those cases would eventually really give us more insights into the more common form of neuroblastoma, where there is not a history of the disease in the family. So the story for us started decades ago, I would say in the late 80s, when we started collecting all of these families where neuroblastoma segregates. So all of these families that have at least 2 individuals with neuroblastoma, and we do not think that that is lightning striking twice. We think that that is because of some underlying genetic underpinning. And this project of trying to use these families where the disease is lethal, you know it firsthand, all of you. And so it's really hard to collect several generation pedigrees. But these are the families that led us to eventually be able to discover ALK. And for me, it started, like I said, in 2000, and this was my project to find the gene that causes the hereditary form of neuroblastoma. And I had the good fortune of talking to this young woman who was 16 years old at the time, and she reached out to me and she said, my sister died of neuroblastoma long ago. I have another sister who is undergoing therapy for neuroblastoma. I have a brother who just has a funny looking eye. And I challenged her to get blood from every member in her family who was still alive. And she got us blood from so many of these individuals in this family. And we were able to use this family to eventually identify mutations in the ALK gene. And these kinds of families are very rare. They're still just about under 3% of all cases of neuroblastoma, but they really are the classic family where there is a cancer predisposition syndrome, where off and on we see the disease come on at an earlier age. Children can have multiple primary tumors. You can see there's incomplete penetrance, and what that means is, you know, you have 2 kids with the disease, you have a mom who clearly is the carrier of what we did not know at the time, but we now know is an ALK mutation, but she's not affected. And then her daughter here is also unaffected. And as we all know, the mortality is very high. So we did this fancy screen back in 2007, actually, that allowed us to see this. So we took the DNA from every single member of every family that we had collected, and we looked to see, was there anywhere in the entire genome, looking from chromosome 1 all the way through last chromosome on the genome, to see, was there some signal that would allow us to look for potentially a disease-causing gene. And there was this signal right here on chromosome 2. And on chromosome 2, what allowed us to narrow the region that ultimately led to the discovery of ALK is this one really unique family right here, where you have a grandmother who was affected with a disease, who had a daughter who had 3 children with neuroblastoma with 2 different dads. So 3 children with neuroblastoma, and we were able to sort of map out here the basically the smallest region of overlap. And we had these informative regions in this one little girl here, and then a more distal region that ended here for this little girl. And it defined for us this region on chromosome 2 that is right here, where there were 104 genes. And in that region was McN, which was kind of incredible. And my mentor, John Maris, said, well, that must mean that McN is the gene that causes the inherited form of neuroblastoma. So I, with my deer in the headlights still, just felt still that I needed to sort of do what I was told. And so I spent 6 months looking for mutations in McN and never found any. And then I said, okay, we need to move on now. So we literally started one by one kind of sequencing these genes based on what we learned about them, and ultimately came up with finding mutations in the constitutional DNA of this oncogene that we now call ALK. And I'll go into this later. So ALK is the major familial neuroblastoma gene. And what we're recognizing now, these were all the families that we published on. But what we're recognizing now is that you don't have to have a family history. We have identified kids who have an ALK mutation in their tumor. And when we test their blood, we find that it's also in their germline. And sometimes that happens what we call de novo. So it did not come from the mom or dad, and sometimes it comes from a mom or a dad who's a silent carrier. Because remember, I talked about this idea of incomplete penetrance. So, what is ALK and how was it originally identified, I think is an amazing story. It started in 1994 with Steve Morris, who was working in Tom Look's lab at St. Jude. And they identified the ALK gene in an anaplastic large cell lymphoma cell line that came from a child who had a rare form of lymphoma that we call ALCL. And we also then started to recognize that these rare sarcomas that we call IMTs, inflammatory myofibroblastic tumors, they also have ALK fusions. And what I mean by a fusion is this is what ALK looks like. This is the membrane of a cell, and part of ALK is on the outside, and that's what we call the extracellular domain of ALK, and part of it is on the inside, and that's where the kinase domain is, and this is where the mutations are. In diseases like ALCL and IMT, what happens is that this whole portion gets cleaved off, and then this part then fuses with another gene that could be anywhere in the entire genome and makes a whole new oncogenic protein. And that's also true for non-small cell lung cancer. So this discovery was made just about a year before our discovery, that about 3 to 5% of patients with non-small cell lung cancer have an ALK fusion. But we were the first to discover point mutations here in the kinase domain of ALK, not only in the germline or constitutional DNA of the families with the disease, but also in 14% of patients with high-risk disease. And mutations now in ALK have become very informative because they've become a mechanism of resistance for patients who are getting ALK inhibition therapy and who have diseases like non-small cell lung cancer and IMTs. We showed that, I'll just really point out the key aspects of the data here. What we did is we sequenced 1,600 tumors from patients in the children's oncology group that we were able to get approval for. And we showed that all the mutations are inside the cell in the kinase domain, that there are these 3 hotspots in yellow. So the majority of mutations are at 1275, 1245, 1174. These are numbers that may mean something to some of you who have children whose tumors have an ALK mutation. We also showed that a subset of patients have not a mutation, but this is what chromosome 2 looks like. And when you have many copies of McN, when McN is amplified, this is what McN looks like. And then down the road from McN, as I showed you earlier, is ALK. And sometimes you can see ALK amplifications. And this is really important. I'll mention later that the Europeans have showed that patients who have this combination of McN and ALK amplification have the absolute worst outcome despite our very intensive therapy. These data show that no matter what, whether you have intermediate risk disease or low risk disease, if the presence of an ALK mutation predicts for an inferior outcome. And we see it in all of the risk groups, in the low, intermediate, and high. And in high risk, we think it's 14%, but I'll show you that I actually think it's a lot higher than that, and we're starting to recognize that. But let me just show you what we did in the lab when we first presented our discovery of ALK mutations in families. I had somebody from Pfizer who we did it in a late-breaking presentation at AACR, and I had one of the chief scientific officers from Pfizer who came over to me and said, "We've got this drug and I'm going to drive it to your lab." And he literally sent it to my lab right after the meeting. We have these cell lines in the lab that represent the mutations that we see in patients. So these numbers are those three hotspots I showed you in yellow. And then the amplified is the one that I showed you with the amplification, this alteration right here. And when we poured this drug, we called it 1066, but it's crizotinib, onto these cell lines, we found that it takes a lot less to kill the cell lines that have a 1275 mutation and amplification. And these cell lines seemed a bit more resistant. And it was the same in animals. So these are animals with neuroblastoma that have a mutation at 1275. When we treat them with crizotinib, we can make the tumors go away. But when there's an 1174 mutation, this may be statistically significant for a statistician, but for me as a clinician, this is nowhere near good enough for a patient. Despite that, we were able to, within a year of our discovery of ALK mutations, open a clinical trial. And that really was thanks to not only all the preclinical work that was done in the laboratory, but the support of Pfizer. So you can see we opened this trial in August of 2009. And we learned a lot from this trial. Ultimately, we defined the dose of crizotinib as this number right here, which is kind of irrelevant. More importantly, it's 200% of the adult maximum tolerated dose. And the rationale for that really came from the science that was being done in parallel that showed that you could potentially overcome the resistance of those mutations if you could expose patients to higher doses of crizotinib. So this really was a home run for patients who have this disease, ALCL. Remember that ALK was discovered in ALCL in 1994. And now Steve Morris calls this the 20-year overnight success story, where you see a patient here, everything in black on this nuclear medicine PET scan is lymphoma. After one cycle, it's literally all gone and it stays gone. These are patients who have prolonged, durable responses, I dare to say cures. And we enrolled 26 such patients. These are their responses, how long they were on therapy. Some of these patients who stayed on therapy only for a short time because their treating physicians felt that they should go on to getting a bone marrow transplant. I don't necessarily know that I agree with that, but those were the decisions made for some of those patients. And these data, just on 26 patients on this phase 1 pediatric trial, provided a breakthrough designation, but from the FDA in May of 2018. And the reason I consult for Pfizer now is that I am helping them to provide the data to the FDA so that there will be a label for crizotinib for these patients. And so that is very exciting. And the same is true for these rare patients who have these sarcomas with these ALK fusions. Similarly, amazing response rates, durable. You can see this little boy had tumor all in his lung, and it just goes away after cycle 3, and it stays away. I mean, these are just incredible responses. And we're gonna get a label from the FDA as well, so that these patients can get access to this drug. The responses for patients with neuroblastoma are really different. And these are data that will hopefully be published soon. This is the neuroblastoma cohort in the crizotinib trial. So what this shows you is the 20 patients, all with ALK mutations. The ALK mutations are defined down here. The only patients who really had some type of objective response were the ones in red who have that mutation at 1275, which is the only one that, again, in our preclinical work, shows some response to crizotinib. And while it seems like the responses are out long time. This is only about a year. The other graphs that I showed you were going out 3, 4, 5, 6, 7 years. There's actually a patient who's been on it for 11 years because we don't know when to stop. And so for the most part, early on, we failed these patients, including Evan, not knowing even that crizotinib did not cross the blood-brain barrier. Even Pfizer did not know that at the time. But we did have proof of concept that ALK is important in neuroblastoma, and that in the patients like this little girl where she has the inherited form of neuroblastoma, really the drug is a home run, because there the mutation in ALK is what drives the disease. It is the tumor initiating event. We met this little girl when she was about 1.5 years old, had refractory disease, 12 rounds of chemotherapy, made 4 surgeries. We thought what was really interesting about her is that when she was born, she had tumors in both adrenal glands, and she had a tumor in the back of her chest. And we thought that perhaps she had a genetic predisposition. She was the first patient that we were able to find in our clinical lab a germline ALK mutation in her constitutional DNA. When we tested her parents, it turned out her mom is the carrier. And I'll just tell you a couple of things about this family because they've taught me so much about ALK. She stayed on the crizotinib trial for 5 years. We didn't know when to take her off, but after 5 years, we took her off, and she's now 6 years out from that. So she's now almost 12. Her mother developed neuroblastoma when she was pregnant with this little boy who is known as Kinsey, who when he was inside his mommy's tummy, we tested we tested part of the chorionic villi, and we knew that he would not be a carrier of the ALK mutation. But then mom developed neuroblastoma, and she is also now hopefully cured, but was on therapy, received crizotinib for 3 years. So we learned a lot of lessons early on in our experience with crizotinib that, ALK is important in many tumors, but turning off ALK in neuroblastoma is much more difficult than it is in diseases where the underlying mechanism is different. And really, we know so little about neuroblastoma biology at the time of relapse, and that's when we're treating patients, right? We're trying to look for how a drug is going to work in patients who've suffered a relapse, who have had so much therapy. And the point I want to make about this slide is that we retrospectively worked with Foundation Medicine to get all of their pediatric data for neuroblastoma to see, because they collect whether the samples that are submitted to them are a diagnosis or a relapse. And what we were able to do is to show that ALK mutations at relapse are much higher. 20% is actually what we published in this paper. That was in 2016, but I can tell you in 2020, it's more like 35%. And to me, that tells me that these mutations are there at diagnosis. We just don't have the technology yet, although we're getting there, to identify them. And that's why I think that the frequency of ALK mutations at diagnosis is so much higher. We had crizotinib. We've moved very far along with crizotinib, and we didn't want to lose the opportunity to provide a targeted therapy for patients with newly diagnosed high-risk neuroblastoma. And so we showed, these are 3 mouse models. I'm only going to focus on this one here, Felix, and I know the whites couldn't be on today, but we'll be looking at this video later. Felix was a very special little boy who we had the privilege of taking care of in Philadelphia when they came from London, because they had found that his tumor had a mutation in ALK at this residue. And we knew that crizotinib, when you treat with crizotinib here, it makes no difference. Chemotherapy here, no difference. When you combine crizotinib with chemotherapy, we see sustained and durable responses. The chemotherapy and the crizotinib are very synergistic. And so with that, we opened in the children's oncology group a phase 3 trial called ANBL 1531. It was activated in the spring of 2018, and it was the very first time that at the time of diagnosis, we are still using very archaic ways of sequencing a tumor. So only patients where the ALK mutation is present in every single cell really are identified. And when they're identified, they're non-randomly assigned to getting our standard therapy in the United States, which includes 2 stem cell transplants and the immunotherapy. They get crizotinib throughout the entire therapy. And when they're done with immunotherapy, we recommended even an additional year of therapy with crizotinib. So this is the current phase 3 trial for children with newly diagnosed high-risk neuroblastoma. And this is what happens right now for patients who are found to have an ALK mutation. In parallel, we're always doing the science, and we think it's really important that there be forward and reverse translation, that the patients really inform what we do in the lab and that the lab informs the clinical trials. So we had studied crizotinib. What I won't tell you about in between is the like 10 other ALK inhibitors that we tested, and they were all like crizotinib, to be very honest. And then we came upon lorlatinib. You can see its structure. It has this ring here, very different than crizotinib. And this is what allows it to penetrate through the blood-brain barrier and to really be effective against patients who have CNS disease. So we were really excited about this drug. And we got even more excited when we tested it in our mouse models that have these different mutations. And I'll again draw your attention just to the one here, Felix, where when this is what crizotinib looked like. Unfortunately, this is all we had when we met Felix, and we knew that it wouldn't work. But this is what happens with lorlatinib. The tumors go away and they stay away. And this is really in memory of Felix here. And we thank you for the gift of being able to have this model to be able to identify new effective drugs. What I want to point out here is that this is the model that has the mutation that we think is sensitive to crizotinib. And look what we learned here. Blue is crizotinib. So we treat these mice who have this mutation, and you see that at about 4 weeks, despite the fact that we continue treatment with crizotinib, the tumors become resistant. But that's not the case with lorlatinib. So we see that it's as effective and even more effective, and the responses are much more durable. With that, we opened a study in the NANT consortium, the New Approaches to Neuroblastoma Therapy Consortium, that is funded by the NCI. And a lot of our work really feeds into this phase 1 consortium to develop drugs. We activated this study in September of 2017. And the drug is taken, it's tablets. You can dissolve them in water once a day, so not twice a day. And it's much better tolerated than crizotinib. And for those of you who have kids who are taking lorlatinib, and I'll give a shout out to a little girl named India who I've never met, but who has touched me very much and who I know is doing really well with the lorlatinib. We really pushed to study many dose levels. We started at 75% of the adult dose, but we're ending up, just like with crizotinib, at 200% of the adult dose. And it's just extremely well tolerated. And we do think that higher exposures are going to lead to better, more durable ALK inhibition in the long term. These are the kids that have been enrolled on the trial, meaning under 18. We also know that there are adults with neuroblastoma, and so we've had a special cohort on this trial, and we presented these data recently at ASCO. We enrolled so far, it's a bit more than 33 patients. Their average age in the kid sort of cohort is 6 years, but it ranged from 2 to 17. And you can see in the adult cohort, look at that, the average age, 22 years. But we've treated and are treating patients ranging from 15 to, I'll now say 52, because this woman has been on lorlatinib for 2 years and is now 52, and she is doing just great. We've seen all the mutations, the relevant mutations in the trial, and we see that the subset of patients who come to us with MYC amplification are the toughest, because at the end of the day, MICN really is this truncal event that makes it very, very difficult to salvage. I'm going to show you what 2 responses look like. This is a little boy with a huge tumor in his right neck that wraps around all kinds of major arteries and veins, impossible to remove. At diagnosis, his MIBG scan here, this is the mass, incredibly hot, very bright. You see it on in a different view here. This is what happens after, I think this was the 4th course. You can see that it's much dimmer. And now after course 10, there's essentially no take up of the MIBG. We biopsy this tissue. The tissue is dead. It's necrotic tissue. It's scar tissue. And this little boy has been on this therapy for it'll be 3 years in February. And actually he was treated at one of the lower dose levels. So we don't necessarily need higher doses to see responses. And then this is what we see in patients who, despite having seen a lot of prior ALK inhibition, this patient is an adult who had seen 2 years of crizotinib, 4 months of alectinib, and this is an MIBG scan, which I'm sure for all of you who have seen these is really kind of chilling, where you see disease everywhere in the spine, in all the bones, in the femurs here. This is what her Foundation Medicine report looked like from biopsy of one of these lesions, where not only is there the ALK mutation, there's also a P53 mutation, which we think is really a mechanism of resistance to therapy. But this patient had a dramatic response. Look at this, after 2 cycles, her Curie score is 0. There's absolutely no neuroblastoma. And I'll share that her response lasted about 2.5 years before the disease unfortunately broke through. We think that lorlatinib in combination with chemotherapy is going to be more effective, and we've shown that in this particular model that has an ALK F1174L mutation and MICN amplification. When you treat with lorlatinib, we see durable responses until about 10 weeks, but unfortunately, despite continued treatment, the tumors do circumvent our treatment. They become smarter, and we're really working hard to figure out why that is. But when you combine with chemotherapy, the responses are really durable. And in the patients who I mentioned earlier who have MICN amplification and ALK amplifications, these are the patients who have the worst outcome. Our European colleagues can tell you that on their trials, the 3-year event-free survival for these patients is less than 20%. And in a model that has these genomic features, what we did is we treated with lorlatinib, chemotherapy, and then lorlatinib and chemotherapy. So this is just the mice that are treated with nothing, vehicle, just like a placebo, basically. In blue is what happens with chemotherapy. So eventually the tumors become resistant to the chemotherapy. And the lorlatinib alone and lorlatinib chemotherapy are overlapping. It's quite amazing. We actually stopped therapy because we figured we're going to be treating forever here at about 18 weeks. And we're actually closer to 40 weeks, and we've not seen any recurrence of tumors. So I really believe this is going to be an effective therapy for patients with these genomic features. And with that, I will end with really the most exciting next phase of ALK and bringing it up front. And this is thanks to Solving Kids Cancer, Solving Kids Cancer Europe. I really want to just give a shout out, especially to Leona and Nick. And I want to thank Pat Tulungan also for inviting me today. We have been granted an award to bring lorlatinib up front in neuroblastoma therapy. And so we're bringing the science to the patients in real time, rather than in the Children's Oncology Group completing our study with crizotinib. The data speaks for itself, and we are calling this collaboration the Titan collaboration. And that's thanks to Nick, who came up with this incredible acronym. And it stands for Transatlantic Integration Targeting ALK in Neuroblastoma. And in the Children's Oncology Group, this amendment will be open in January. And I know that my colleagues in SIOPEN, who I speak with multiple times a week, are working very hard in the current HRNBL2 trial to open the arm for patients with ALK alterations. We will be using a method of next generation sequencing to allow us to identify as many patients as we possibly can to get lorlatinib. And the lorlatinib will continue after immunotherapy for another 12 months. And thanks to all of the support that we've had from Gavin and from the Evan Foundation, we'll be performing these liquid biopsies throughout therapy to really monitor for response and to understand what the drug is doing. I'm going to end there and just thank the many people in my lab and all of my collaborators and funding sources, especially Solving Kids Cancer and the Evan Foundation and so many others. These are some of the incredible kids I've had the privilege to be part of their life. Here's Evan. This makes me smile. This was just such a great moment. And Felix here. So many other special kids. And then I want to just show you the members of the Titan collaboration in the Children's Oncology Group, in the SIOPEN, along with Pfizer, who has been incredibly supportive and anticipates that if there is the activity we expect to see, that these data will go to the FDA for a label indication. And I want to, again, thank you today, Solving Kids Cancer from both the UK and the US, and the Children's Neuroblastoma Cancer Foundation. Thank you very much for your time, and I'm happy to take any questions.
Wow, Yael, thank you so much. As I said at the beginning, what a great story of progress and hope, right? And you and everyone at CHOP just works tirelessly on this, and we're in your debt. And you know, the ability that you have to do this science, but also have an endless reserve of empathy for families is unique and special, and just blessed to have you here in the neuroblastoma world. And we topped out at about 72 folks. I know there were little technical glitches along the way for some, but yeah, we had over 70 people, still 67 folks with us. And we have about 12 minutes or so for questions. So let me jump into it quickly. There are 6, Yael.
Okay.
You're going to have to help me a little bit with some of the scientific language here. Let me see if I can just get us started with the first one. Is ALK positive, and then in parentheses, weak but convincing cytoplasmic staining different from ALK mutation? And then the question goes on to read, is it the ALK expression in neuroblastoma cells, which is present in most neuroblastoma cells?
Yeah, that's a really important question and important distinction to make. The vast majority of neuroblastomas do express ALK on the surface of the cell. And that's really exciting because we're developing actually immunotherapies to target ALK when it's expressed on the cell surface. But unfortunately, ALK expression does not correlate or does not predict for an ALK mutation. The vast majority of neuroblastomas have ALK expression. And so if you're told that your child's tumor has ALK expression, that doesn't mean that there's a mutation. The tumor needs to undergo sequencing at the DNA level to be able to determine whether or not there's an ALK mutation. So expression is not a surrogate for mutation.
Got it. Thank you. And then a few people are interested in this question. Can you discuss the side effects from the 3rd generation of ALK inhibitors, lorlatinib?
Absolutely. So lorlatinib is extremely well tolerated, and I know that crizotinib isn't. That's been really tough for kids and for families. I can tell you from our experience on our phase 3 trial, and lorlatinib is a tablet that you take once a day. It can be crushed and dissolved in water. The main side effect is weight gain. It's actually an on-target effect, meaning it's because it goes into the blood-brain barrier through it, which is great for neuroblastoma. We need that. But it plays with the control center for appetite. And so it is direct effect of an increased appetite that leads to an increase in weight. For the kids, that's actually a blessing. For the adults, it's not. For whatever reason, it causes an increase in cholesterol that we don't understand, but that's something that we can easily manage. And I would say that a lot of parents say when their kids are on lorlatinib, they just feel good. They have this kind of high energy,
Wow.
which is also a really, positive sort of side effect. The other side effects that are described, the neuropathies, the swelling of the legs, the cognitive changes, the changes in memory, and these are not seen in kids. They're seen in adults for sure. We have not seen them in kids. And we are doing very close neurologic testing with a psychiatrist who's part of our team so that hopefully we can show the FDA that this drug is safe for kids.
Terrific. So, yeah, we're going to enter into the lightning round here. We have 5 minutes left. So, FOX2B, if I'm pronouncing that correctly, can immunochemistry show FOX2B positive and then become negative after induction treatment?
No, FOX2B really should remain positive. It is just, it's what we call the core regulatory circuitry of neuroblastoma. So live neuroblastoma cells will always express FOX2B.
Great, thank you. Next one up, regarding ALK amplification in the 4% of high-risk neuroblastoma, do you see a difference in response rates to ALK inhibitors between mutated versus amplified?
Well, in our preclinical models, we see that ALK amplification is exquisitely sensitive. And I'll have to tell you that I've had to convince a lot of people of that because there are many who think that based on the adult studies that amplification is a marker of resistance, but it's not. And I really believe, and I hope I'm right, that the kids with ALK amplification are going to have dramatic responses to lorlatinib, and that there'll be significant improvement in their survival.
All right, let's go quickly here to the next one. About the different percentage of ALK incidence at diagnosis and relapse. How was the detection run? Was the liquid biopsy run as well as double check?
Yeah, great question. We are developing these very deep sequencing assays to be able to identify patients who have mutations down to 1% of their genome, and these patients will be eligible for getting the ALK inhibitor lorlatinib on both the European and the American study. We've been working really hard to develop these assays, and we follow them with liquid biopsies to see if they come back, if what happens at relapse. But it is really important, I think, to get these kids, to find these kids at the beginning, right? The goal is cure from the beginning, not to have to go through everything so many of you have been through, and then have ALK come back, or if it's something else, but often it's ALK, and then have to start again.
Here's a question specific to the UK. Does Dr. Mosse know what the current amendment is to lorlatinib trial at the Royal Marsden as it is currently paused?
Yeah, that really frustrates me. And I know it frustrates a lot of my colleagues at Solving Kids Cancer who've been supporting this. I don't know why it's paused. It shouldn't be. It should be open. I know the Curie has had a really hard time opening it for reasons I don't really understand, to be honest, but it should be open at the Royal Marsden. And if you know of a patient who needs access to lorlatinib, I will help you get access to it and get you on the trial or get you the drug separately.
Great. Just a minute left here. Yael, low risk ALK positive, surgery to remove 2 primary tumors. Is there a higher risk of relapse?
No.
No. All right, that's a good answer.
Yeah.
Next question. Has lorlatinib showed any positive effect on non-ALK, non-MYC cases?
It's not necessarily MYC dependent at all, but in non-ALK, lorlatinib so far is not effective, and we've looked at that. So it's really got to be patients who have activation of ALK through either mutation or amplification.
Great. So let me just wrap it up here. That's all the questions. There's other comments here, Yael, about you and how wonderful you are, which I concur with. And there's a shout out to the funders of the Titan study for their incredible support of your work in this trial. So, hey, let me just close it up here right at the 12:15 mark my time, and say thank you again. There are obviously additional sessions that are teed up now. We have one on 8H9 for CNS relapse and PTSD and post-traumatic growth after childhood cancer. So please, if you're interested, head back over to the Watch Live page on the event website to begin viewing. And remember, this has been recorded, so if you want to revisit this, you can go to the on-demand page on the event website. Thank you so much, Yael. Thank you, everyone, for joining us. Have a great conference. Bye-bye.
Thank you so much, Gavin, and thanks to everyone. You guys take care and stay safe and be well.
Thank you.
Bye-bye.